All right, guys, we'll just give a minute to get some more people joining us here. Okay, well, sort of a crappy day out there for a Friday, but that's all right, I guess. Get a couple dominoes set up here just to demonstrate one last part. I need to talk about the threshold and the all or none response, which is our next thing. First off, you guys have handed in your Neuron worksheet number 1. I haven't had a chance to get that marked, and your Neuron worksheet number 2 was due at noon today. Those will be marked both and back to you. And then after class today, I'll share a third one. So the third one kind of goes through some of the concepts that we're going to talk about today. And so I've decided that we'll have the quiz. The third one will be due Tuesday at noon, and then your quiz will open Tuesday. So it'll open on a different day than it normally does. And we'll go from Tuesday to Thursday for the Neuron quiz. But on Monday, we're going to start looking at the brain, the spinal cord, and kind of the branches of our nervous system. Okay? Questions before we get started? Okay, I don't see any questions, so we will move right into going into the worksheet or into the PowerPoint here. All right, so the all or none response. Put that there. We'll start from this slide. Okay. So last class we talked about threshold level. So threshold level is the minimum level of stimulus we need in order to have a neural transmission occur. Okay, so the minimum stimulus we need in order for that wave of depolarization to occur. That's our thresholds. We have to meet threshold or we don't get that wave of depolarization. All right, so that ties into what we call the all or none response. So the all or none response means that neurons and effectors, so remember we're talking about anything that's being affected by a motor neuron. So that can be, a gland, or anything that's being stimulated by a motor neuron also has an all or none response. So it's not just neurons, it's motor neurons as well, okay? So neurons and effectors will either respond to stimuli with complete neural transmission if the threshold potential is reached or exceeds, or not at all. Okay, so those are the only two things that can happen. Okay, so either it'll respond completely or it won't respond at all. Okay, so we either get a wave of depolarization or we get nothing. We never get a faster wave or a slower wave. It doesn't matter how much stimulus there is. If it's just met the threshold just by a teeny tiny increment, or if it's well exceeded the threshold, it doesn't matter. It'll respond all the way or not at all. Okay. All right. So let's take a look at our, this is going to be hard to see here. Let's take a look at our dominoes again. Okay. We'll try to get these dominoes back to my main screen here. Hold on one second. Oops. There we are. And let's just stop sharing. Sorry guys, I'm still getting used to my just want to be just get rid of this. There we go. All right, and stop share. There we go. There's me, and there is my dominoes. Okay. So here's my dominoes. Okay, so now we can see my dominoes nicely lined up there, sort of at an angle. All right, so we talked about threshold last class. So threshold, if I just barely tap this first domino, right, it wiggles a little bit, but we don't have a wave of depolarization. So I haven't met threshold. If I, sure these are lined up okay, if I touch the domino and I touch it enough, just barely touch it, but enough to reach the threshold, I get a wave of depolarization. If I touch it a lot, so I just wham into that, so hit it really hard, whoops, see that didn't demonstrate right now, hit it really hard, I get the same wave of depolarization, right? So it doesn't matter whether I've totally exceeded the threshold or if I've just barely gone over the threshold, we get the same response. It doesn't cause that wave of depolarization to go faster or slower with more or less intense stimulus. So the neuron will either respond completely or not at all. That's the all or none response. Okay. All right, so let's go back to our screen here, because there should be a question in your head right now as I'm talking about this. Right? And that question should be, okay, so that's all fine and good. A neuron can either respond all the way or it doesn't respond at all, except for the fact that we detect intensity of things. We can detect when a light is bright or dim, when a sound is loud or soft, when someone barely touches us or touches us with like a lot of force, like a punch, right? We can tell the difference. So we can detect intensity. We can tell if something's warm or super hot. But a neuron can only respond one way. So we still need a way in order to detect intensity. So all or none happens. That's how it works, right? But here's how we detect intensity. There's two different ways. So intensity, how do we perceive intensity? Well, the first way we perceive it is by how many impulses are stimulated, how many neurons are stimulated that are reaching their action potential, right? So if I think about this, so I'm just going to get out my whiteboard here. Find it. Okay. Share the whiteboard here with you for a second. So if I think about this, right, I'll make a little graph. So this is number of neurons that are affected. Okay. Because we have to remember, neurons have different threshold levels, right? Some have really low threshold levels, so any stimulus is going to cause a wave of depolarization. And some have very high, so it's going to take a really intense stimulus, okay? So if I have increased intensity, basically what that means is that more neurons are going to be affected, right? So the more intense my stimulus, the more neurons are going to be affected. And that's where my brain's going to perceive it. Okay, more neurons are affected. I know that this is a bright light. I know that someone hit me hard. I know that this is hot, right? More neurons have reached the threshold level. If it's less intense, then it's only going to affect the neurons that have a really low threshold. But if it's super intense, it's going to affect the neurons with a low threshold, medium threshold, and high threshold. So more neurons will be stimulated, okay? So that's one way that we can detect intensity, right? Okay. The other way we detect intensity is by variations in frequency. So basically what that means is a neuron refires again more quickly. The wave of depolarization still either happens or doesn't happen, happens at the same speed every time, but how frequently it's re-stimulated increases. And that again, if it re-stimulates more often, that signal is going to go to my brain. This is more intense stimuli, okay? So those are the two things that lead to us being able to detect intensity, because every neuron has an all or none response. But we have to remember in our body, we have multiple neurons. So that's where we are talking about this response here, right? Because we have multiple neurons. So our number of impulses that we get because more neurons are reaching their threshold, and then variations in frequency, how often they're stimulated. Okay? Any questions about that? All right, so we'll move on here. Okay, this video here, we're not going to watch because it's about 28 minutes long. I would strongly recommend watching it. I steal quite a few things out of this video to try to teach because they have a lot of descriptors in it. And sort of examples. But nonetheless, it's not a bad thing to kind of sit and watch. And what else are you going to do on a Friday night, right? During a pandemic. Okay. Our next concept is called synapse. Now, we've talked at this point about neurons being stimulated and then what happens. But we haven't talked about what causes neurons to become stimulated, right? They get stimulated, sodium channels open, yada, 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 right? But now we're going to talk about what actually leads to that occurring. So when we use this term synapse, it's referring to the message that's being sent either between one neuron and another neuron, okay, or between a neuron and an effector. So that's the synapse, and that's where our neural transmission, so our wave of depolarization, is going to get converted into a chemical message. Okay. All right. So neurons are separated. So remember I told you when we talked about having our dendrites and cell body and axon, and then that's going to send a signal to the next neuron. There's a gap there. And that's a little teeny tiny gap, but it's called the synaptic cleft. Okay, so that gap is called the synaptic cleft. It's very small, so it's about 20 nanometers. A nanometer is a billionth of a meter. So it's a millionth of a millimeter. So it's a teeny tiny little distance. And that's how far apart they are. So on a regular light microscope, you wouldn't be able to have the resolving power to be able to see that distance. Okay. All right, so let's take a look at what's going on. So I'm just going to slide this stuff down so we can see the top part of this diagram to start off with. All right, so here, if we look on the left-hand side, we're looking at two different neurons. So here's the first neuron. So this is the axon of the sending neuron. This is the neuron that's sending a message. And then this is the receiving neuron. All right, so the neuron that's sending a message we call the presynaptic neuron. So pre means before, that prefix, right? Before synapse. So that's the neuron that is before the synapse. So it's going to send the message. And then our next one is the postsynaptic neuron. That's the neuron that's after the synapse. So it's going to receive the message. Okay. Now, sometimes this isn't a neuron, right? If we're talking about a motor neuron is sending a message to muscle or glands, in which case we would refer to this as a postsynaptic membrane rather than a neuron, okay? All right, so if you look at this little box that's been highlighted here, that little box is what we see right here, okay? So this box is an expanded area. In this box, what we see is we see the synaptic knob. So there's the very end of one of these branches off of the axon, okay? And we see this action potential, so that wave of depolarization, it's gone through the dendrites, the cell body, and it's making its way through the axon, right to the very bottom part through the synaptic knob. So there we see the synaptic knob, right? Okay, so what happens? These little ball shapes that we see here are called synaptic vesicles. Okay, so remember from science 10, vesicles are basically little structures, little organelles that kind of store stuff and move it around the cell. So they just have stuff in them. They're little sacks of storage, okay? So here's our vesicles. Inside the vesicles are a chemical called a neurotransmitter, and these guys are super important, okay? So these vesicles are filled with neurotransmitters. What happens is as that action potential moves its way down the synapse, or down the axon, it forces these synaptic vesicles to fuse with the end of the synaptic knob. And as they fuse, they open up, and through exocytosis, they dump all of their materials into this gap, the synaptic cleft. So now the synaptic cleft is filled with the neurotransmitters. Okay. All right. So then what happens? I'm going to move these little things to the top now so we can see the bottom part of this diagram. Okay, there we go. So now what happens is if we take a look at this little square here, that's what's being expanded at the bottom here. Those neurotransmitters attach to receptor sites. The receptor sites are on ion channels. When it attaches to a receptor site, it opens an ion channel. Okay? Well, let's take a look specifically at this neurotransmitter. This neurotransmitter is attached, and all of a sudden we see ions flowing into the postsynaptic neuron. What diffusion channel causes ions to flow into the neuron? Remember, we talked about two different diffusion channels. What diffusion channel causes ions to move into the neuron? What diffusion channel? Yes, the sodium diffusion channel. The sodium diffusion channel causes ions to move into the neuron. So that's what we see happening here, is these are sodium ions moving into. So that's going to cause the postsynaptic membrane to become depolarized. If enough sodium comes in, then it'll reach threshold and we will get another wave of depolarization, which will go through the next neuron. Okay? The next step we need to look at is oftentimes these neurotransmitters are broken down because we can't just have them sitting here forever. Because if they sit in the synaptic cleft forever, they will continue to reattach to the receptors, continue to open up, continue to cause depolarization to happen. Okay, so they need to be broken down. So enzymes usually come along and they will break down neurotransmitters. And there's a particular neurotransmitter and a particular enzyme that we need to know the name of that we'll get to in a minute. Okay. All right. So that's just a quick summary of what happens in synapse. Okay. So if I'm looking here, I'm seeing the sodium channels are opening, so sodium's diffusing into the postsynaptic neuron. Okay. All right. Okay, so synapse. So we'll just have a couple of words here. So the neuron carrying the impulse to the synapse is the presynaptic neuron. Okay. It causes, that action potential causes the synaptic vesicles, which are filled with neurotransmitters, to fuse with the membrane at the end of the synaptic knob. Synaptic vesicles release neurotransmitters into the synaptic cleft. Neurotransmitters diffuse across the synaptic cleft. Okay. All right, so now we're at the postsynaptic neuron. So the neurotransmitters attach to the specific receptors on the membrane of the postsynaptic neuron or the cell of the effector, right? So remember, it can be a neuron or an effector that has these receptor sites. The receptors are found on the diffusion channels. So there's actually receptors on potassium diffusion channels and sodium diffusion channels. Depending on the neurotransmitter, either sodium or potassium diffusion channels will open. Up to this point, we've only really talked about sodium channels opening. Okay, so sodium or potassium diffusion channels will open. Okay, so we have two types of neurotransmitters. We have stimulatory or excitatory neurotransmitters. These are the ones that cause sodium diffusion channels to open. So we can say stimulatory sodium, right? Those two go together. They both start with S. So stimulatory neurotransmitters cause the sodium channels to open, which means we have that wave or that quick diffusion of sodium ions into the cell and the membrane becomes depolarized. Okay, so depolarization occurs in the postsynaptic membrane. If we have enough sodium, we reach threshold, we get a wave of depolarization. Okay, but this other one is a little bit different. These guys are inhibitory neurotransmitters. They cause more potassium channels to open. Okay, so let's think about this. If we have a membrane and our membrane is polarized, okay? So we have our pump that's always pumping. It's always pumping sodium to the outside and it's always pumping potassium to the inside, right? And it's polarized, so our sodium channels are closed. So that's our sodium channels. But our potassium channels, some of them are open, right? When it's polarized. So some potassium is diffusing out. But when we have an inhibitory neurotransmitter, what happens is they attach to those closed potassium channels and they cause them to open up. So now we get more potassium on the outside and we end up with too many positives on the outside of our membrane and negative on the inside. So what's that state when there's too many positives on the outside? What ISD word do I use to describe that when there's too many positives on the outside? We saw this word before. Too much potassium is getting to the outside. Excellent, thank you. It becomes hyperpolarized, too much positive, okay? Now this is referred to as an inhibitory neurotransmitter. So we know when a neuron's recovering, it overshoots, and we have that tiny moment where that graph dips and we have hyperpolarization. But we can also have inhibitory neurotransmitters that cause this to happen. So we ask ourselves, well, why do we want this to happen? And probably the best example, it happens through our brain, but one of the best examples is when we think about our muscle groups, right? So in that reflex video, we saw that. Because if we want to do a bicep, right, work our biceps, every time my bicep contracts, my tricep has to relax, right? So that means I need stimulatory neurotransmitters to stimulate my bicep, but I need inhibitory neurotransmitters sending signals to my muscles of my tricep, okay? So that's probably the best way for us to really understand what inhibitory neurotransmitters do. So when we think about this in terms of my analogy, so let's just go through, just gonna open these up. I'm just gonna go through what my analogy has been with this stuff. So, let's see if I can do this. There we go. Okay, let's move this back for 1 second. So remember we talked about how polarized is our resting state, right? So when we're polarized, we're resting. And then when we get stimulated, it's depolarized, right? And then we're recovering, getting back to polarized. So I can still use a similar analogy to talk about hyperpolarized, because let's think about my arms at my side, and let's imagine that being hyperpolarized is that I have a 50-pound weight in each of my hands. Okay? So that means that I'm inhibited, because no matter what stimulus now hits me, I can't overcome that to depolarize. So it is now inhibited, okay? So that's where we can use that analogy of our hands raising and falling, is we want to think about that as being two 50-pound weights, one in each hand, and it's making it so it's almost impossible for that neuron to be stimulated, okay? So that's inhibitory. So the outside of that cell becomes more positive than a resting potential, and therefore depolarization can't occur. So that's inhibiting depolarization. Okay, so we have a couple little videos that I want to watch in regards to synapse. So I'm just going to clear this and go to this. All right, so this first video, actually the first two, they talk a little bit about calcium ion channels. While calcium ion channels do play a role in this process, they are not in the curriculum for us. So you can kind of just gloss over that part. One day if you take this in university, you'll have to know that, but not for Bio30, okay? So we'll just watch this little video here. Oh, whoops, sorry, just 1 second. I forgot to share my sound with you. Let's try that again.
Along the neuron to the axon terminal as an action potential.
Neurotransmitter molecules are manufactured by the neuron, stored in synaptic vesicles, and transported to the axon terminal. To initiate synaptic transmission, an action potential must reach the axon terminal. Arrival of the action potential at the axon terminal causes voltage-gated calcium ion channels to open. The opening of these channels allows calcium ions to flow down their concentration gradient into the axon terminal. Arrival of calcium ions in the axon terminal causes synaptic vesicles to migrate to the presynaptic membrane. Synaptic vesicles fuse with the presynaptic membrane, releasing neurotransmitter molecules contained within the synaptic vesicles to the synaptic cleft by the process of exocytosis. Neurotransmitter molecules diffuse across the synaptic cleft. The postsynaptic. The processed pieces of neurotransmitter molecules are reabsorbed into
All right. Then I have another one here that we'll watch.
And cause synaptic vesicles to release.
We'll just go back here.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive.
Okay, I'm just going to pause it there. So this is talking about a particular neurotransmitter. There's lots of them, but this one is called acetylcholine. Now, acetylcholine is causing sodium ions to go in to the postsynaptic neuron. So I would like you to tell me, is acetylcholine a stimulatory neurotransmitter or an inhibitory neurotransmitter? Is it a stimulatory neurotransmitter or an inhibitory neurotransmitter? Okay, it is stimulatory, S for sodium, stimulatory. Perfect. Okay, let's go back to this.
If the membrane potential reaches threshold level, an action potential will be produced.
All right. Oh, there's anything more to that. All right, next we have
Synaptic terminal.
Oh, stop. Next we have one more video here, and this one is my most favorite one. It's just a really good illustration of what happens with a neural transmission. Okay, so it's a nice 3D model here. So we'll just get this out of the way here. And okay, whoops. So what we see there is this wave of depolarization with a wave of repolarization right after. So a wave of depolarization as those positive ions are rushing in, and then a wave of repolarization as the positive ions are rushing out. So remember that rush of positive ions going in, what kind of ions are rushing in? What is that, a potassium or sodium that are rushing in with that wave of depolarization that we see? Is it sodium or potassium? So during that wave of depolarization, is it sodium ions or potassium ions? Yeah, it's sodium ions are rushing in, right? So we see that recovery happening right after, and that's kind of why I really like this, is to get to see that. So we'll just play that one more time. Right? Rushing in, coming out, rushing in and coming out. Okay? Net positive on the inside, net positive on the outside. Okay, so now we're at the synapse. Okay. pause it here for 1 second. Okay, so we're sitting at the synapse here, and I have the benefit of being able to do this. I'm going to just get my pen out. Actually, let's get my spotlight out. Okay, so first of all, we're just going to review what we did, see how everyone's understanding this. I see these little ball shapes. What do we call these little ball shapes? What are those called? Okay, that whole structure, what do I call that? Synoptic blank. Anyone remember? The synoptic knob would actually be this whole big shape right here. Vesicles, synoptic vesicles. Okay, so now I want to just look here. So what do I call the gap here? What's this gap called? Good, the synoptic cleft. So our gap is the synoptic cleft, okay? All right, so now, these little red dots that I see here, what are the little red dots? Okay, I see them up here and I see them here. What are those? Little red dots. What type of chemical are those? Those are the neurotransmitters, exactly. And what do we think these purple dots are? What are the purple dots? Purple dots are sodium. And I know they're sodium and not potassium because they're going into the postsynaptic neuron. Okay, all right, so let's finish this off. So if enough sodium comes in and it reaches threshold, then that next neuron will have a wave of depolarization. All right, so we'll go back to the PowerPoint here. There is one more on drug use. We may or may not get to that today. It's not critical for us to understand anything to do that one. So we'll see. Okay, so let's take a look at this image here. And just get my spell it again. Okay, so again, we want to just go through this process. So we have the nerve impulse, right? And as the nerve impulse comes down, it's causing these synaptic vesicles to fuse with the bottom of the synaptic knob. And they're going to dump their neurotransmitters into the synaptic cleft. Okay. The receptor sites are on the ion channels. So if we take a look here, right, the neurotransmitter went on this receptor site, causing sodium ions to go in. So we know this is a sodium ion channel. So we know in this case, the neurotransmitter is a stimulatory or excitatory neurotransmitter because sodium ions are going in. Okay. All right. So it's a stimulatory neurotransmitter because sodium ions are moving into the cell. Okay, so now let's take a look at this. So we're going to go through some of these questions here. And again, I can't remember what order I put my animation, so whatever. Okay, so if I look at the words at the bottom here, these are the words that are going to go in my blank. I have lines here that indicate some of these things. So here's all of my words, my word list. And then I've got one here and one here and one here. So on the chat, I'd like you to tell me what goes where. So let's start with the very top here. Thinking of the words that I have in my word list, and it's synoptic. I have this covered up here. Synoptic vesicle. Okay, so screen's too small. Synoptic cleft, synoptic knob, and synoptic vesicle. Okay, so what's this? What is this pointing at? All right, good. So we're looking at the dendrites. Okay, and now again, thinking of the word list, what are these ones pointed at? So look at the word list. Okay, we're looking at axons. And looking at our word list, what are these guys? Synoptic knobs. All right, let me move these up now so we can see what's on the bottom here. Okay, so now let's look at the image here to the right. And we'll just, so this line here, again, looking at the word list, what's this line pointed at? So it's pointed to right here. What are we pointed at? Since we've already chosen synoptic knobs, this one here, it's actually pointing to right to this surface right there. So we're looking at the presynaptic membrane. Yeah, it's hard to tell where it exactly is, where I'm meaning for it to be pointed to. Okay, this one is going to this surface right here. So what would that be? Okay, so it's pointing right to this point right here. Awesome, so that's our postsynaptic membrane. Okay, this one's pointed to this region right here, where my little spotlight is. There. Okay, good. The synaptic cleft, excellent. All right, now this one is pointed to this big ball here. Good. Vesicle. All right, now this one's pointed to the little balls inside the big ball. Okay, little balls here inside the big ball. Neurotransmitters, excellent. And then the last one is referring to this little structure that's sitting here. Yeah, and those are our receptors, which of course are ion channels that have a spot for the neurotransmitters on them. Awesome. So make sure you know those structures and know what they do. Okay. All right. The last main concept, well, we have a couple of things left to do here, is summation. Here's the problem, right? At the very beginning, I said that thousands of neurons can communicate with each other. But I've also told you that a neuron can only respond in one way. It either completely responds or does not respond at all. Okay, that's it. There's only, or sorry, respond in two ways. Nothing or everything. That's it. So what I'm looking at here is I'm seeing this neuron, okay, and I'm seeing dendrites of this neuron. And then I'm seeing that a lot of presynaptic neurons are attached to it. And presynaptic neurons are attached that are giving excitatory or stimulatory neurotransmitters. And some are attached that are giving inhibitory neurotransmitters. So the red ones are inhibitory and the green ones are stimulatory. Well, what's a postsynaptic neuron to do? Does it react? Does it not react? So this gets us to the next concept of summation. Summation means that all the effects of all the presynaptic neurons are added together, all the pluses, stimulations, and all the minuses, inhibitions, all come together to yield one response. That is summation. Summation is the net effect of all of those presynaptic neurons because our postsynaptic neuron can only respond in one way. Okay? So postsynaptic neurons can only have one response, either complete neural transmission or no neural transmission. Okay? Now, the no neural transmission could be that they're inhibited. The no neural transmission could be because the excitatory and inhibitory have balanced, right? And they're just nothing. It could be either one of those, but no transmission occurs. Okay. All right, so let's take a look at this example here. So I pulled this out of an old textbook. But here I see I've got 3 presynaptic neurons. Neuron A, neuron B, and neuron C are presynaptic neurons. Neuron D is a postsynaptic neuron. Okay, we're measuring what's going on in neuron D. All right, so if I look at what's going on in neuron D, when neuron A, so not B or C, when just A, so cross these guys out for a second, when just A is stimulating D, does it yield an action potential when I look at it? Do I see an action potential? Does D have a wave of depolarization when it's just stimulated by A? So if I take a look here, we can see that neuron D, when it's just stimulated by A, doesn't have an action potential, right? A little bit happens, a little bit of sodium ion channels open, and I see that they open because this goes in a more positive direction, but I don't have this peak, right? I don't see a wave. It doesn't meet a threshold level. Okay, so if I look at B, is B a stimulatory or inhibitory neurotransmitter? We can just write an I or an S, inhibitory or stimulatory neurotransmitter for B. Yeah, B is stimulatory. Stimulatory, I know it's getting more positive, but it just doesn't reach threshold, so I don't get that huge influx of sodium ions, right? What about C? Is C stimulatory or inhibitory? Yeah, so C causes more potassium channels to open, which means they get even more positive on the outside. The outside becomes hyperpolarized, right? Okay. But what this does is this shows us what the impact of summation is, because when we take A and B and put them together, A by itself just doesn't have enough impact. It doesn't open up enough sodium channels. But if A and B both stimulate D, it causes more sodium channels to open that actually reach threshold and cause a wave of depolarization. And now a signal is going to go through D. Okay. So if I look at the side here, neuron A, because it causes our membrane potential to become more positive, we know that it's stimulatory, but it doesn't reach threshold. Okay. C, because it reduces the membrane potential, we know is inhibitory. Okay. And then D, I said this word already, but go back here. The stimulation of neurons A and B causes an action potential in D. The combined effect on neuron D is known as, and that's a word that we need to know, is summation, right? So when we take that net effect of the neurons that are impacting a postsynaptic neuron, we call that summation. We add them all together, okay? All right. Next. Okay. Our last thing to talk about is types of neurotransmitters. Okay, just gonna move this up again so we can. So there are 2 neurotransmitters you need to know the name of. The first one is acetylcholine. Acetylcholine is found in the brain and it sends messages between neurons. Okay, so the shortened form for acetylcholine is ACH. It's also used by motor neurons to send messages to muscle cells. So it goes from neuron to neuron, but acetylcholine is also affects our muscle cells. So it's in a motor neuron. Almost always, acetylcholine is a stimulatory neurotransmitter, which means it goes to receptor sites that are on sodium ion channels. Okay, so sodium ion channels open in the postsynaptic neuron. If enough open to reach threshold, then we get a wave of depolarization. Okay. All right. Cholinesterase. So I said there's enzymes that break down neurotransmitters. Cholinesterase is the only one that you need to know. It is an enzyme that breaks down acetylcholine. Some insecticides and nerve gases, so an example of a nerve gas like this is sarin, blocks the release of cholinesterase. Now this is a problem, okay? So when we think about, let me just put up my whiteboard here. Okay. When we think about this, so here's my, whoops, sorry. Here's my synapse, right? There's my synaptic knob. There's my synaptic cleft. And I'm going to just draw A's for acetylcholine. So I've got all of these A's sitting here, and they're going to come to these receptor sites, oops, that are going to allow sodium in, right? If they attach to these sites. And here's another sodium ion channel that lets sodium in, oops, whatever. And sodium goes in, and we have these acetylcholines. Well, what's supposed to happen is cholinesterase is supposed to come along, shoot. Undo. There we go. Cholinesterase is supposed to come along, and it's supposed to get rid of these acetylcholines. So that's going to stop those ion channels from being opened, right? But if cholinesterase is inhibited, then we have all of these acetylcholines that are attaching to these ion channels, and we just tons of acetylcholine sitting in the synaptic cleft. So as soon as it's done its job, it's going to come off, right? When it's done its job, it's going to come off. But then very quickly, another one's going to go right back on. And it's just going to repeatedly cause depolarization, recovery, depolarization, recovery, depolarization. And there's very little opportunity for full recovery. Okay, so let's think about what the impact of that is. So if we go back to the PowerPoint here. Okay, so basically the synaptic cleft is now just filled with acetylcholine, and there's no way to get rid of it. When we think about our little ants, so if you look at your little bottle of that white powder used to kill ants, it actually usually says cholinesterase inhibitor, so it blocks cholinesterase. Well, for these little ants, their little ant muscles get their signals from acetylcholine. And if cholinesterase doesn't take it away, those muscles are constantly going to be stimulated, and they're kind of in this state of paralysis. Okay? Sarin gas, which was used by the president of Syria on his own people, is a nerve gas that does the same thing except to human beings. Okay? Causes a buildup of acetylcholine in the synaptic cleft, so it's stimulating those muscle cells, right? So really what can happen, because our muscle cells, okay, if we're paralyzed, we can't walk, but if our intercostal muscles can't move and our diaphragm can't move, we die. We can't breathe, okay? So totally deadly gas. Okay, norepinephrine is used in our sympathetic nervous system, so it sends messages through our sympathetic nervous system to help stimulate our fight or flight response, okay? It is not a hormone. Well, it is a hormone as well. So there's a hormone and a neurotransmitter. They're both called norepinephrine. Okay, really quickly, we're just going to end up with fill in the blank here. So I know we're getting past our date here by 5 minutes already, but let's just quickly go through the fill in the blank. All right, the gap between two neurons, okay, not between Schwann cells, but between two neurons is called what? Excellent, the synaptic cleft. In the synaptic knob, neurotransmitters are stored in. So what structure are they stored in within the synaptic knob? Vesicles. Good. Our neurotransmitters are in the synaptic vesicles. Excellent. Okay. Receptors for neurotransmitters are found on the pre or post synaptic membrane. Which one? Post. Good. Stimulatory neurotransmitters cause what ion channels to open? Sodium or potassium? Excellent. Sodium, stimulatory. Inhibitory neurotransmitters cause what ion channels to open? Potassium. All right, good job. All right, next one here. Over here. Oh, I just told you the answer, shoot. Inhibitory neurotransmitters cause the postsynaptic membrane to become hyperpolarized. Hyperpolarized. All right, let's back up. Summation refers to the collective effect of all of the blank neurons on the blank neuron. All right, just use pre and post so you don't have to type the whole thing for your answers. Okay, so collective effect on all the presynaptic neurons have on the, what's the second blank? Postsynaptic neuron. Good. All right. Acetylcholine is a neurotransmitter that blank neurons use to stimulate muscle cells. So motor or sensory? Yeah, motor for muscle, M and M. Cholinesterase is an enzyme used to break down. What does it break down? Enzyme used to break down. Acetylcholine. Good job. Okay. Epinephrine is a neurotransmitter that is used by what branch of the nervous system? Excellent. Sympathetic branch of the nervous system. Okay. All right, guys, so that's it for today. I do have a video or a little activity I think we'll do on Monday on muscle cell, or just drugs a little bit on how they affect neurotransmitters, and then we'll talk about brain. Okay, so you have a quiz on Tuesday. I will post your next worksheet that'll help with the stuff that we've done today and hopefully get you all understanding that. Probably before 5 o'clock today you'll have that, so that'll be due Monday. Your quiz will be Tuesday. All right. If you have any questions, just throw them in the chat, or don't forget to email me if you have anything. And we will see you guys Monday. Enjoy your weekend.